All right, let's dive in. Today, we are cracking the code on the parts of genetics that trip up almost everybody. We're talking about the exceptions to Mendel's rules that are practically guaranteed to pop up on your CUET exam.
We all learn about Gregor Mendel, right? The father of genetics. But what if I told you his groundbreaking rules were really just part one of the story? Let's get into what happens when inheritance patterns get, well, a whole lot more interesting. And why does this matter so much to you? Because, look, this isn't just theory. We're talking 2 to 4 direct questions from this topic alone on your CUET exam. Getting this stuff down isn't optional. It's absolutely essential if you want a top score. So you're going to want to stay focused.
So we all start with Mendel's really simple idea: dominance. One gene wins, the other one, it just hides. Pretty straightforward, right? But then scientists started doing more experiments, and this one particular flower came along and just completely shattered the rules, opening up a reality that was way more fascinating. That's right, it all came down to one small pink flower. According to the simple dominant recessive rules, this shouldn't even happen. This single little anomaly, the snapdragon, basically forced us into a whole new understanding of how genes are actually expressed. So what's going on when alleles decide not to play by those old rules? Well, this brings us to 2 concepts that are absolutely critical for you to know: incomplete dominance and codominance. Let's break them down right now.
Okay, first up, incomplete dominance. The key word you need to burn into your brain here is blending. It's like mixing paint. With snapdragons, the dominant allele for red isn't strong enough to completely cover up the recessive one for white. So when you cross a pure red flower with a pure white one, you don't get red, you get this beautiful in-between phenotype, pink.
Now, here's the crucial point for your exam: this ratio. When you self-cross those pink flowers from the F1 generation, the next generation gives you a phenotypic ratio of 1 red to 2 pink to 1 white. See what happened there? The phenotypic ratio is exactly the same as the genotypic ratio. This is a huge Q-trap. So many students just automatically write 3 to 1. But for incomplete dominance, you have to remember, it is always 1 to 2 to 1. Okay, let's clear up the single most common point of confusion. For incomplete dominance, remember the keyword blend or in between. Think red and white paint making pink. Got it? For codominance, the keyword is coexist or together. Both alleles show up fully and equally. The best example is AB blood type, where you have markers for both A and B. They don't blend, they both appear. Now, let's take this a step further. Mendel's peas only had 2 options for each gene. But in the real world, what if there are more than 2 choices? This brings us to a really cool concept called multiple alleles. Multiple alleles just means that for one single gene, there are more than 2 versions of it floating around in the population. The classic example, of course, is the human ABO blood group system. It's controlled by the gene I, but there are 3 alleles out there: IA, IB, and little I. But, and this is super important, even if there are a dozen options in the population, any single one of us can only have 2 of them, right? One from each parent. And this table just shows so brilliantly how all these ideas work together. Take a look. Blood type A, that can come from 2 different genotypes. Same for B. Type O is purely recessive, easy enough. But look at blood type AB. The genotype is IA-IB. This is where multiple alleles and codominance totally meet. The A and B alleles are codominant with each other, but both of them are completely dominant over that little recessive I allele. It's all happening at once. Understanding all this isn't just about passing an exam. It has real-world, sometimes life-or-death consequences. Let's look at how these genetic patterns play out in medicine and, well, even survival itself. Take the Rh factor in our blood. This actually brings us right back to simple Mendelian genetics. Rh positive is dominant and Rh negative is recessive. The genetics might be simple, but its importance is massive, especially during pregnancy, where an Rh negative mother carrying an Rh positive baby can lead to really serious complications. And now for a bit of a darker twist: lethal alleles. These are exactly what they sound like: genetic combinations that are fatal. The classic example is coat color in mice. The allele for a yellow coat is dominant. But if a mouse embryo inherits 2 copies of this dominant yellow allele, it's a lethal combination. That embryo just won't survive. So what's the big clue for a lethal allele on your exam? It's this ratio right here, 2 to 1.
If you cross two heterozygous yellow mice, you'd normally expect a 3-to-1 phenotypic ratio. But because those homozygous dominant ones don't survive, you're left with a ratio of 2 yellow mice to 1 non-yellow mouse. If you see a 2-to-1 ratio in a question, a little alarm should go off in your head that says lethal allele.
Okay, time to lock in these critical numbers. Seriously, think of these ratios as your secret weapon. They will help you instantly decode almost any genetics question that the CUET throws at you. Let's drill this. You see a 3-1 ratio? That's your classic straightforward Mendelian complete dominance. You see a 1-2-1 ratio? That screams incomplete dominance. And if you see a 2-to-1 ratio, that's your signal. A lethal allele is in play. Burn these into your memory right now. All right, enough theory. It's time to see if you can actually apply this stuff under a bit of pressure. I'm going to throw some exam-style questions at you. I want you to pause for each one, try to solve it, and then we'll go over the answer together. You ready? Let's do this. First question. Pink flowers and snapdragons are a result of A, B, C, or D? Go ahead and pause now and really think about it. And the answer is B, incomplete dominance. What was our keyword? Pink is the blend, the in-between phenotype from the red and white parents. It's as simple as that. Okay, let's try a trickier one, an assertion-reason question. The assertion is, ABO blood group shows both codominance and multiple alleles. The reason is, there are 3 alleles for this gene in the population. Think about the relationship between those two statements. And the answer is, both are correct, and the reason is the correct explanation for the assertion. Think about it. The very fact that there are 3 alleles, that's multiple allelism, is precisely why it's possible for the IA and IB alleles to exist together and show codominance in someone with AB blood type. Before we wrap up, let's quickly go over the biggest traps. Number 1, do not confuse blending with coexisting. Number 2, the ratio for incomplete dominance is 1-to-2-to-1, not 3-to-1. Number 3, an individual can only carry 2 alleles. Period, no matter how many exist in the population. And number 4, a 2-to-1 ratio is a massive clue that a lethal allele is involved. If you can avoid these, you are way ahead of the game. And I'm going to leave you with one last challenge to work out on your own. If a person with genotype IA has a child with a person of genotype IB, what faction of their children could possibly have type O blood? Work out that Punnett square and drop your answer down in the comments below. This is exactly the kind of problem you're going to face. Thanks for joining me.